We'll start today. The what you call soul of this chapter now. That what is chromosome and what are the genes and what is the factor allele that is being discovered or started initiated by George Gregor Mendel. So we today are going to see a chromosomal theory of inheritance. मतलब ये क्रोमोसोम्स नेक्स्ट जनरेशन में जाते कैसे हैं और नए नए फिनोटाइप्स एक्सप्रेस होते कैसे हैं या फिर जो पैरेंटल कैरेक्टर्स हैं वो ऑफस्प्रिंग में और उसके नेक्स्ट प्रोजेनी में हाउ दे आर बीइंग ट्रांसफर्ड दैट इज इनहेरिटेंस and this is being inherited by a specific chromosome like others are autosome and those others which participate in the next generation expression of gender that is allosomes. Okay, so before going into that how chromosomes go ahead, first we focus on the landmark work of John Gregor Mendel. Okay, his work was almost beyond the limit of excellence considering the era. See, the era where he worked on Pisum sativum, the genetical work was 1865. There was no any kind of technology or advancement in the life that we people are fortunate to have. But in that era also, his statistical analysis and interpretation of the result and that record keeping was beyond and someone's imagination in that era, but still having such a blunder effect of experiment, his theory was remain unrecognized for almost 30-40 years. So there were some controversial fact or stigmas associated with his work at that time. We'll see one by one. Just two, three are the major one. First one, what was Mendel's concept that the factors which transferred from parent to the offspring, only that factor will express. That means if there was tall and dwarf, so there will be only tall or dwarf in the next generation. There will not be an intermediate or incomplete dominance, that which we have seen in Snapdragon or Antihynum majus. Okay, so first thing that he was not what you call supporting the blending of genes or incomplete dominance. Second fact, he was the first person who introduced mathematical record keeping in biological study and analysis. So it was not digestible to the biologist of that era. Of course, the thinking changes with the generations, and in that generation, biology was considered as a unique than the mathematics as its own unique. So interdisciplinary science study was not initiated. Okay, and what he called factor or allele that transfers from parent to the offspring or next generation, he had not any physical evidence for that. Of course, the F1 he showed how dominance affect. F2 he showed how segregation occurs, independent assortment occurs, and the new combination come to the existence. But what is that factor and where it reside? He has no clues or physical evidence for that. And because of these, what you call considerable limitations, his work wasn't got a good what you call flash in that era. But his work was so outstanding and records were so outstanding. Then three scientists independently from different countries, Holland and Germany, in the year 1900, they rediscovered and redid the work of Mendel. They were Hugo de Barris, is a very famous person, Karl Kurens and von Mark from Germany. Okay, so these three person they redid the work of Mendel and come to the same conclusion. What Mendel come to the conclusion after their work was redid by these scientists. At that time, the microscopy was slightly advanced, not much advanced like today, like electron microscope or else, but still, microscopic techniques were almost developed. Staining techniques were developed. So these staining techniques can be a boon as a cytological study, and all the cytologists means those who study the structure and function of cell and its organelle, they were doing a remarkable work with the help of microscopy, which was not possible at the time of Mendel's work. Okay, now the cytologists have their share of opinion to see how the cell functions. Beside that cell, how the nucleus operates. Inside that nucleus, chromosomes do exist, and these chromosomes show a specific existence and characteristic in embryonic. So before embryonic stage, at the time of embryonic stage, then at the time of what, a reproductive phase, they produces gamete, and during that gamete production they undergo a very specific and unique process, that is meiosis, which is divided into two parts, meiosis 1 and meiosis 2. All that study of chromosomal behavior was possible by cytological study. Now the concept or the conclusions of Mendel can be justified by direct physical evidence existing in the chromosome as a gene, to which Mendel called as factor. Okay, now by doing cytological analysis, two scientists, one from America, one from Germany, certain genetics from America, who was studying the chromosomal behavior in case of grasshopper, all male and female grasshopper, and their chromosomal existence as well as behavior during mitosis and meiosis. So cell division and this gamete production was almost being studied simultaneously by German scientist Boveri also. And Boveri, he studied in sea urchin. He said that for proper development of the embryo, all the chromosome must be present, that is obviously from both the parent, all the chromosome must be present for the embryo development. So independently they studied very nicely, and they combined their theories of chromosomal, what you call, segregation and inheritance. So they together compiled their work, and they, what you call, put forward the theory of chromosomal inheritance. Now what were their conclusion as a cytological study by taking the behavior of chromosome and genes, and what was the challenging task to interpret or correlate with Mendel's work? We'll see in 3 points. Okay, first of all, Sutton and Boveri, they concluded that chromosomes comes in pair as homologous. Now what is homologous? Homologous means two chromosomes, that means one chromosome which are in pair. Now both these chromosomes are of equal length, as well as stain alike, as well as the location of genes present on both the chromosome is alike. So what is Sutton and Boveri's conclusion? First, that chromosomes comes in pair, that means they are homologous. What was Mendel's conclusion? That factors comes in pair. That was his, what you call, first preference to come towards the dominance, that factors comes in pair, out of which one dominates the other, so one become dominant, other become recessive. Second fact, chromosomes segregate during meiosis. Of course, during gamete formation meiosis occurs, because they studied the cytological analysis of the chromosome in the nucleus. So chromosomes segregate during meiosis. Now meiosis is in two parts, 1 and 2, that we'll see. But just correlate, what was Mendel's concept? That law of segregation of allele during gamete formation. These allele segregate, and they reach how much, what he said, 50% from each parent. So during gamete formation, the allele segregate, which were in pair, that is in their allelic form. And here what Sutton and Boveri concluded, chromosomes segregate, separate during meiosis. Obviously they separate. And what was Mendel's correlation there? Laws of segregation, that is alleles segregate during gamete formation. Now Sutton's and Boveri's final blow. Different chromosomes assort independently, that will study by the behavior of chromosome and pairing formation. You recall the dihybrid and the monohybrid cross which we have seen. That in case of that dihybrid which we have seen, that yellow and what you call, round and yellow, wrinkled and green. In F1 generation, all were round and yellow. But in F2 generation, we got round, green, as well as yellow, wrinkled. That means that R, round, and capital Y, which we have seen, was not always associated with each other. they segregated independently and showed a new phenotype, and that is the application of independent assortment of the gamete. They brings the new phenotypes, that is the raw material as a variation for evolution. So what is the third conclusion? That different chromosomes assorts independently. So you recall that dihybrid cross formation of that green with round and then wrinkled with yellow, they were possible because of independent assortment. And what was Mendel's work here? Law of independent assortment, that what we have studied in the dihybrid cross. That means allele segregate and express independent of their what you call counterpart allele or the genes. Now, in this way, if we just directly express this conclusion how chromosome behaves, just we have to consider the example of this meiotic behavior in the chromosome and its inheritance to the next cell. Okay, now see, we know meiosis is divided into two parts, meiosis 1 and 2, and in these two we have two times metaphase, metaphase 1 and metaphase 2, and we know at the time of metaphase all the chromosomes align at the metaphasic plate, and in anaphase they move apart to the opposite pole, both in anaphase 1 and anaphase 2. Remember one thing, in case of meiosis 1 or anaphase 1, homologous chromosome move apart from that metaphasic plate. Okay, so these are just to refer given the alphabet for their color. See, these are homologous chromosomes. Okay, O for orange, Y for yellow is associated orange with green and yellow with red. This is one possibility. Okay, in another possibility, at metaphasic plate combination can change. The orange may get associated with red and yellow may get associated with small green one. Okay, so these are the possibility 1 and possibility 2. Both these possibilities are occurring at what time? At that metaphasic plate during anaphase, when they are moving to the opposite pole. This larger chromosome will get associated with shorter one. So this orange with green or this orange with red, that are two possibilities. Okay, so what happens in meiosis 1, anaphase 1? That homologous chromosome separate. So we got separated homologous chromosomes. Here orange with green and this yellow with red, one possibility. Orange with red and yellow with green, another possibility. What is separated here? The homologous chromosomes are separated. Okay, now once these homologous chromosomes are separated, in meiosis 2 we know that it ends with formation of 4 haploid gametes. Now here you see, after this meiosis 2, what we got 4 gametes from this one diploid cell after meiosis 1 and meiosis 2. Meiosis 1 was reduction division and meiosis 2 is equational. Okay, now what we are getting here, these 4 haploid gametes, that this orange can form gamete with green also, and here this orange can form the combination with red also. What does this means? That law of segregation, the chromosome segregate. Different chromosomes assert independently. Different chromosome asserts independently. Orange green can be possible, orange red can be possible, and this is possible at what time? At anaphase 2 and metaphase 2. The combination may separate and a larger and small chromosome can assert with different perspective. Either orange can go with green or orange can go with red. Same way yellow can go with red and yellow can go with green. Where it is possible? Remember, at metaphase 1 and 2, all these chromosomes align at metaphasic plate. So which combination comes at that metaphasic plate and how they get replicated, what you call move to the opposite pole, is just a chance phenomena. So what does this means? That these chromosomes are not always stick with each other. They assort independently, and because they assort independently, they carry independent another genes. That's why we get new combinations and new phenotypes. But this is just a mere explanation, although it was scientific correct and everything. To have this as a conclusive explanation, one more American scientist give the what you call final blow to this chromosomal theory of inheritance. What he did, Thomas Hunt Morgan, the father of experimental genetics. You just imagine a banana and you remove its peel and keep as it is. What will happen to that banana? It start to degrade obviously, and if it start to degrade, you see some fruit flies coming out there and buzzing. It may look yak to observe, but these are the fruit flies which are being used as a landmark tool in genetics to study the behavior of chromosome. Even the sex-linked inheritance was also being started by studying these Drosophilla melanogaster fruit flies. Now, how he studied was also what you call a dramatic sequences there. He was studying these fruit flies, and he kept all his fruit flies in the vials, different vials. One day suddenly what he saw, that all those fruit flies were of red eyes. Okay, so one thing to remember, those fruit flies which he was studying were of red eyes. One day suddenly he got in those vials where a lot of fruit flies are there, one of the fruit fly has developed a white eye, and that was shocking. So what this T.H. Morgan with his associate collaborators, what he did Mendelian experiment with a red eye Drosophilla and with the newly appeared white-eyed Drosophilla. Now what he considered, everyone there was red. So he considered a red eye as the dominant fruit fly, and the white, which is one, which just what you call a few in number, he considered that as what you call recessive. And what he did, he crossed. How he crossed? Just like we have crossed the tall and dwarf, what you call Pisum satirum. Okay, so he crossed the red eye Drosophila with white eye. Of course, what we get in the F1, capital R and small r. Obviously, you can guess now what will be the color of the eyes in F1 generation of those fruit flies. Definitely it will be red. Go to the F2. Of course, what is the F2? When we do selfing of F1, so capital R small r is crossed with capital R small r. Now just put that into checkerboard and Punnett square. What we will get? This combination will get RR in the first place, both capital. Second, heterozygous capital R small r. Third, heterozygous capital R small r. And fourth, small r small r. Now here, what was the ratio? See, one homozygous red, two heterozygous red, and one homozygous white. That means three phenotypes as red, one phenotype as white. Out of these three, one was homozygous red and these two were heterozygous red. That means you can see the ratio genotype, one homozygous red, two heterozygous red, and one homozygous white. And that is the F2 ratio of Mendel. That is 1:2:1. And what was the phenotypic ratio? 3:1. 3 red as to 1 white. Now that gave the final blow. Yes, chromosome inherit in that way. What the Sutton and Boveri come to the conclusion, which was the basis, came from the actual work of the great father of the genetics, that is Mendel, that what he called factor, these factor were in pair. Of course, these were pair in means they were on homologous chromosome, and that means what they considered as factor, we call them as genes. And yes, genes do have same logo on the different chromosome because chromosomes are homologous in nature, and during segregation or gamete formation, each chromosome comes equally from parent, one from mother, one from father. And what happens, we know that here in interphase the amount of chromosome content is doubled. Obviously meiosis I, that is reduction division, homologous chromosomes segregate, and it comes into two daughter cell. Because these content were doubled in interphase, now these homologous chromosomes are no more homologous, they are now sister chromatids. They separate from each other and they becomes 4 haploid gametes. Any combination is possible in the metaphase and metaphasic plate arrangement and anaphasic separation to the opposite pole. So in this way you can just read the NCERT content once or twice, see the diagrams there of orange, green and yellow, red, and see the work of what they have done, that is Sutton and Boveri. You can just formulate this conclusive point: what are chromosome, what are homologous chromosome, what are allele, what are genes in pair, that is Mendel's factors, and what is segregation and what is independent assortment. If you correlate this with that dihybrid cross or monohybrid cross and how that new phenotype occurs by that independent assortment, you will come to the same conclusion what T.H. Morgan came to the conclusion in that Drosophilla, melanogaster, red eye, white eye, F1 heterozygous red, and in F2 both homozygous red, heterozygous red, and homozygous white. So just read those diagrams in NCERT once or twice, read the content and see the video once or twice. You will definitely understand the very interesting and easy topic, that is chromosomal theory of inheritance. So I hope you understood this. So thank you. Thanks for watching.